we start to talk about cultivating microbes. And the process of microbial cultivation consists of what is called 5 I's, meaning the letter I. It's incubate. Don't try to write down all 5s. You will see them. We will go through each step. Inoculation, incubation, isolation, inspection, and identification. And we're going to talk about inoculation first. That's the first step. So first of all, what is inoculation? Inoculation is placing a microorganism into some type of the growth medium. When you collected samples from your palms and swabbed the agar plates, you inoculated agar plates. When you will collect samples from the environment and inoculate the plates next week, you will do inoculation. Does that make sense? I want you to be able to distinguish between inoculation and infection. You can infect. Result of infection is in the live organism. You can infect me. You cannot infect a plate. Is that clear? Also, you can inoculate me, but it may not necessarily lead to infection. If you inoculate me with something and this something doesn't replicate in me, there's no infection, just the act of inoculation. Make sense? Clear? So the first step to any procedure of microbial cultivation is to collect the sample. And the most important part of that process is to use the sterile instrument. Okay? Makes sense, right? Now, environmental samples can be any. What are the common clinical samples that are used in clinical microbiology? What is collected from patients? In blood. What else? Urine, great. Stool, feces, yes. Saliva, yes. No, air. I mean, sometimes people are asked to cough, but that's essentially mucus from the respiratory tract. Sweat, I would say skin sample rather than. Sweat by itself, when sweat leaves the sweat gland, it's sterile. I'm just curious, there's one fluid that nobody. You can, yes, you can do. It's more of a, snot is not that convenient. Nasal swabs. Yes, you can collect nasal throat swabs. It's one really important fluid. Well, they're all secretions, but like exocrine secretions. But there's fluid, like biological fluid. Sperm can be collected. Sperm can be collected, mostly collected in case of if STD is suspected, then all secretion, penile or vaginal secretions will be collected. Yeah. Amniotic is sterile if it's not sorry, but the fetus is screwed. CSF? Yes. CSF. I would put CSF in the same line with blood and urine. In people who come with something that reminds meningitis or encephalitis, if lumbar puncture can be done, that's the first thing to do, because CSF must be sterile. And if it's not, it's bad.
What is CSF?
Cerebrospinal fluid. That's
Okay.
what is done during the spinal tap, the collection of cerebrospinal fluid. Does that make sense? All of this should be taken sterilely to avoid any potential contamination. Now, when we inoculate medium can be different. Okay, there are many classifications, and the first classification of the medium is based on its physical state. So we're going to start with the one that you already encountered. Solid medium. Agar that you used to grow microorganisms is an example, the solid medium. It has solid surface. And what is important about agar is that it allows you to isolate, separate microorganisms based on the colony appearance. Remember when you did your hand microbes? You could see white and yellow microbes. You could separate them and grow them as individual microorganisms. Does that make sense? Isolate microorganisms because it allows them to grow in colonies. Agar is not really suitable medium though to accumulate large volumes of microorganisms because come on, how many colonies are you going to grow? It's not a lot. If you want to grow, and important feature of agar itself is liquefiable, meaning it's liquid at the high temperature and solid at the low temperature. So first of all, I don't know if you knew, but you can buy agar in the grocery store. It is used to make jello and other like jello type stuff. It costs a fortune, like a little tiny can, like 10 or 15 bucks. But yes, you can buy agar in the grocery store. Agar is produced from some algae, tropical algae. You can eat it. So I mean, that stuff that's on the plates, technically, you can eat it. You're not going to die. It's not toxic. It's going to taste like crap, but it's not toxic. Okay? So you boil it, it becomes liquid. And you can pour it in the plates, and it'll become solid. This is really convenient because you can boil the agar, and when it cools down, first of all, you can autoclave it. You can sterilize it. Does that make sense? And then when it cools down, you can add to it certain chemicals that can't be heated. Say you can add antibiotics to it. If you will boil the agar with antibiotics, antibiotics will all, disassemble, okay, decompose. But you can add it to agar that is cooler. It solidifies at about the temperature of 50 degrees Celsius. I don't remember what it's in Fahrenheit. Sorry, something like 120 something Fahrenheit. Now, liquid medium here is water-based, and we call it liquid because it's liquid at room temperature, basically. Can contain various types of nutrient supplements like extract of beef or extract of milk or yeast, something that has proteins, fats, carbohydrates. Liquid medium uses for two main purposes. One, to grow huge amounts of bacteria. You can inoculate like a pint of medium, well, scientifically half a liter, okay? And you will have a load of bacteria, a lot of microorganisms, okay? Another use for liquid medium is the indicator medium. For instance, this on the left is uninoculated, negative control. Two media on the right are bacteria. They are inoculated with bacteria that ferment. Okay? When bacteria ferment sugar, they produce acid. And this acid drops the pH of the solution, changing the color progressively to yellow, from orange to yellow. Does that make sense? That's convenient. You cannot separate different microorganisms. If you accidentally inoculate liquid medium with more than one species, you won't be able to tell them apart, obviously. Does that make sense? Now, this is semi-solid. Semi-solid, best characteristic that I could find, it's a clot. So this way of pouring the medium into a tube is often called slant. Okay, because it's inclined. Why we call it semi-solid? If you will flip the tube and hang it after some time, that semi-solid medium will slowly move down. Does that make sense? So it's not completely solid, but it's not liquid either. Okay? Now, what do we use semi-solid medium for? First, we can use it to determine bacterial motility. The word motility refers to movement. Imagine that you have this slant, and you collect some of bacteria at the tip of, say, a really long needle or a wire, and you pierce this slant with a wire. So far, are you following me? If bacteria is non-motile, cannot move, it will grow in the slant exactly where the piercing went through. Does that make sense? Exactly where the wire went through. If it's motile, it will start, so if this is wire coming through, bacteria will start to spread to the sides. Does that make sense? Producing this spread growth. So this is motile bacteria. That is really convenient way to address the motility. Am I clear? But perhaps more frequently, semi-solid medium is used to figure out different types of microorganisms using indicator media. Now this is an excellent example here. On the right is the negative control. Okay? That makes sense? On the left is microorganism that can ferment two sugars, glucose and lactose. So since it, and this medium here, I forgot to mention, is called triple sugar iron agar. The name, which is a tone breaker, tells you that this medium contains 3 sugars and iron. So here, this is E. coli. E. coli can ferment both glucose and lactose. It produces a lot of acid, so the entire medium changes its color to yellow because pH drops. Does that make sense? You see this empty space here at the bottom of the slant? This is CO2. Because during fermentation, carbon dioxide is produced. This, I believe, is Enterobacter aerogenes. It ferments only glucose, so it does not produce as much acid, and acid is mainly concentrated at the bottom of the tube, and there is no CO2. This, one of two, it's either Salmonella typhi or Proteus mirabilis. These microorganisms produce gas known as hydrogen sulfide, H2S. H2S interacts with iron cations, leading to the formation of iron sulfide, which has very characteristic black appearance. So using these slants, you can distinguish between 3 different bacteria based on the appearance of the slant. This is for people actually made a whole array of various semi-solid media in the tubes, and by which slant changed the color and how you can identify precisely what kind of bacteria it is. We're going to do this. We're going to do this lab. We're going to try to identify microorganisms. Yes?
What did you say about the second soup from the left?
Second is only glucose fermenter, so it doesn't produce as much acid and does not produce CO2. So there's no separation at the bottom, and the amount of acid is much lower and concentrated mainly down there in the tube. Enterobacter aerogenes. Now, to make you feel better, you do not need to memorize any of the examples. Am I clear? Type of questions that I'm going to ask you about this. I give you a description of a medium. I tell you, for instance, it is not, has no solid surface, looks like a clot. You tell me that's semi-solid. Does that make sense? I will not ask you about any examples. What I can do, I can describe the medium in a lot of details, and you will have to give it names, terms. Are we clear? I will elaborate on that in a little bit. Any questions about this part? Types of media based on composition and purpose. So composition, media can be synthetic or media can be complex. For synthetic medium, chemical composition is known. We know exactly how many chemicals and what type of chemicals we put in that medium. Does that make sense to you? So say I use sodium chloride, glucose, 21 amino acid to make a medium. I put each component on the scale. I know exactly how much of each component goes into the solution. I know the exact chemical composition. Am I clear? In complex, it says at least one component is not chemically definable. What do we mean by that? Let's say you make a chicken stock. Do you know the exact chemical composition of chicken stock? No, you don't. Can you grow bacteria on chicken stock? Sure, absolutely. Okay? Does that make sense? You take soy protein and just take soybeans, isolate protein from it, break it down with some enzymes. Do you know the exact chemical composition? How many of each amino acid is there? No, you don't. So that is complex medium. Media like TSA, trypticase, soy agar that you use. We're going to use blood agar. We're going to use saburot agar. All these agars, they usually have undefinable component. So they are complex. Now, when would you use each of those? You would use synthetic for the organisms with no nutritional needs, meaning you know that this bacteria requires so much of this amino acid, so much of that amino acid, so much of glucose, so much of, I don't know, calcium chloride. Does that make sense? Complex media will be used to isolate microorganisms that you know nothing about. Think about that TSA agar that you used. It has a breakdown of soy protein. So that's why trypticase soy protein. It has some yeast extract. It has agar. So it has a lot of nutrients. When we put a microorganism on this agar, we don't know what this microorganism needs. But we have a pretty good feeling about the fact that agar will contain all the necessary nutrients. Does that make sense? Think about complex medium as a buffet and defined medium as a fixed menu. So if you come to the restaurant which doesn't serve gluten-free and you are gluten intolerant, you have nothing to eat, right? Does that make sense? You might ask, wait a minute, but complex media is so much easier to work with. It definitely will provide nutrients for the microorganism, right? Turns out, the defined or synthetic medium gives you a pretty neat tool to figure out what are the roles for different chemicals. in the growth of a particular microbe. Let's say you make synthetic medium. And then you say, you know what? In my experiment, I'm not going to add amino acid alanine. And I will see if microorganism will grow in this medium. And if it does, you say, well, apparently this microbe can make its own alanine. And then you say, now I'm going to throw away tryptophan. And microbe doesn't grow. And you say, okay, microorganism cannot make tryptophan. I need to add it. Does that make sense to you? The whole idea. We clear? Good? You understand the idea between complex and defined? Purpose. General purpose medium. Trypticase soy agar. What we're going to be using. Mixture of nutrients. What else can I say? It's like, it's a buffet. Does that make sense? General purpose. Supports the growth of wide variety organisms. Gives no preference to any organism at all. We clear? Enriched. Enriched medium contains some kind of a nutrient that is required by fastidious microbes. Now, what is fastidious microbe? Picky microbe. The great example of fastidious microbe is Neisseria. You may never heard the genus name, but I bet you've heard the names like gonorrhea and meningococcus. Both of these diseases caused by different species of Neisseria. Meningococcus is Neisseria meningitis, and gonorrhea is Neisseria gonorrhea. Turns out that these microorganisms prefer to grow on the chocolate agar. It doesn't have chocolate. It has cooked blood. And what do you think these organisms require from the blood? They need iron. They need much more iron that is usually provided in the regular medium. Does that make sense? So Neisseria is fastidious bacteria, and chocolate agar is enriched medium. Clear? Selective. Medium that inhibits growth of some microbes while allowing others to grow. Is this concept, does it make sense? Say, there's an agar called Mannitol salt agar. This agar contains a lot of salt. The majority of microbes cannot grow in this type of medium. We good? So, let me finish with differential and I'll give you an example. Differential medium. Differential medium allows many different microorganisms to grow, but when they grow, they look different. You understand what I'm trying to say? So, some colonies can be pink, other colonies can be white. Some microbes can make medium yellow, some microbes do not make medium yellow. So you can tell them apart. They're all going to grow, but you can tell them apart. Are we clear? Does that make sense? Kind of question. I give you a description of medium. This medium, blah, 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 allows only fungi to grow. What kind is that? So that's selective, because it selects for fungi. Does that make sense? This medium allows all microbes to grow, but they all look different on it. What kind of medium that is? Differential. Clear? There's going to be several questions of this sort. Now, in the last few minutes, few words about different types of cultures. Pure is the culture that consists of only one species. Makes sense, right? So what you had today your culture, the Serratia culture, that was pure culture. Okay? Mixed is your culture from last week from your hands, because you had different species. And what's important, you knew you're going to have different species, right? And finally, contaminated. Contaminated culture is when you hope to have one species, but you end up with more than one. Somebody spit in your culture, or something else terrible happened. Sometimes contaminated culture leads to the discoveries. Penicillin was discovered after Alexander Fleming saw fungus-contaminated culture of Staphylococcus aureus. It was pure contamination. It was like he didn't wish for it. He managed to, figure it out. You see the difference between these three? Now, for viruses, what is the medium? Viruses are intracellular pathogens. You have to grow viruses in cells. Here on the right, you see images of cell culture vessels. Each flask, you can see, has a layer of red fluid. This red fluid is the special medium that allows mammalian cells, or any other cells, to grow on the bottom of that flask. So if you would take this flask, put it under the microscope, and look down there, you will see many cells growing on the surface. Does that make sense? It may sound really trivial, the cell culture, but if you think about it, the only alternative to cell culture, to growing cells in the flask, is to use animals, which makes it really expensive. I cannot. So, for example, vaccine against yellow fever was produced after Max Taylor infected mice, and he infected mice, sacrificed them, collected the virus, infected second batch of mice, sacrificed them, collected the virus. He did 200 passages. God knows how many mice he slaughtered. Okay? At those times, mice were probably cheaper. Now, it's going to be a golden project, okay, to grow something in animals. And what if the virus replicates only in monkeys? That would be even worse, cost prohibitive. What if it replicates only in humans? Then it becomes really sure, right? Unless you're Dr. Mengele. So, Renato Dulbecco developed the cell culture in 50s, I want to say, 40s or 50s. And that he got Nobel Prize for it. Rightfully so, because that was a groundbreaking discovery of a new tool that allowed virology to take off. Not only virology, a lot of other studies. Does that make sense? We're going to wrap it up now. You want to